When we talk about pathogenic microbes, we can describe three different general types of pathogens. The first is opportunistic, the second is obligate, and the third is accidental. An opportunistic pathogen is a pathogen that can be transmitted without causing disease, but will cause disease under certain circumstances with the host. In other words, an opportunistic pathogen can be thought of as taking advantage of an unusual opportunity to cause disease. That's very different from an obligate pathogen, which is always pathogenic anytime it's transmitted to a host. It must cause disease. An accidental pathogen normally does not cause disease at all in the host, but only accidentally. So, an opportunistic pathogen is taking advantage of certain circumstances within the host that ends up causing disease. These include things like a weakened immune system, an altered microbiome, and what are considered to be breached barriers. There are natural barriers to the entry of pathogens into the body and the development of disease. In other words, there are natural barriers that prevent a pathogen inside the body from going on to cause disease. A lot of times these barriers become broken when the host is suffering from another disease. That allows that pathogen, which might otherwise have just sat in the body and not caused any harm, to go on and cause disease. Obligate pathogens, remember, are always disease causing in the host. In fact, they have to cause disease in order to be spread, in order to be transmitted from one host to another. If you think about Mycobacterium tuberculosis, the person who gets this pathogen and develops TB will then go on to spread that TB through the disease, through the coughing that accompanies the disease. A good example of an accidental pathogen is the bacterium called Neisseria meningitidis. This is actually an organism that's considered part of the normal flora in the nasal cavity. But under certain unusual conditions, the barrier that exists between the back of the nasal cavity and the brain becomes breached. It becomes broken down slightly. And that will allow this organism that normally doesn't cause any problem to gain access to the meninges. And when it gains access to the meninges, it will cause disease. That's an accidental pathogen. There's another one called Bacteroides fragilis that is part of the normal flora in the colon. And this organism, again, normally doesn't cause the host any problem. But under certain circumstances, it will accidentally get exposure to a part of the body where it will cause disease. And this typically happens after surgery, penetrating surgery, and certain penetrating traumas. The organism gets out of the colon and into the blood supply and causes sepsis. So where exactly do pathogens come from? Are pathogens always pathogens, or do they somehow develop across time? As we've said, sometimes pathogens come out of the normal flora. They come out of the microbiome. Sometimes an organism that has existed in a symbiosis with the host for long periods of time will develop a new virulence factor and become pathogenic. That development would be the result of some kind of a mutation in the organism. The good news about pathogens that arise out of the microbiome is that because they resided in the host organism prior to that point, the host has some immune memory of that organism. So these don't tend to become the worst of the pathogens. Another origin term that you'll hear is this word zoonotic, and that word just refers to an animal. So a zoonotic pathogen is one that comes from an animal source. And again, this happens through mutation events, where the pathogen that originates in one particular animal species mutates and gains new traits that allow it to be transmitted to humans. The organism that is causing the current pandemic, the coronavirus, is believed to have zoonotic origins. It's believed to have arisen in a bat species and then undergone a mutation event that allows it to now infect human beings. Last on the list is this term novel, and that word just means new. So there are pathogens that are brand new in the sense that mutation events occur that change their genome, that alter their genes enough to give them new traits and allow them to infect a new host. The difference then between a zoonotic pathogen and any other type of novel pathogen is just where the organism emerged. If it emerged from an animal host, it could be both zoonotic and novel. And the new coronavirus is an example of that. But there are other times when novel pathogens arise that didn't emerge from another animal, but rather from a human in one form. And now they are novel. Now they have new genes and new traits and are pathogenic in another form, but still in humans. The biggest problem with a novel pathogen is that because it has this new genetic material, because it has new genes inside of it and new traits from those genes, it enters the body as a brand new microbe. In other words, there is no immune memory to that organism. That's why during a pandemic, like the current pandemic, everyone is susceptible to the pathogen all at once. Nobody has been exposed to this new pathogen, so no one has any immune memory to it. The other thing I'd like to discuss today is some terminology around the source of pathogens. The term reservoir is applied when we're talking about where a pathogen normally resides and multiplies. Now, a reservoir is a source of infection for susceptible hosts. Sometimes there are living reservoirs. In other words, sometimes pathogens normally reside and multiply in living things and cause either no disease at all or subclinical disease. Remember, we said subclinical disease is infection where the host organism has such a low level of symptoms they don't realize that they have the disease. They don't realize that they're carrying and transmitting the pathogen. So humans and other animals and even plants can be reservoirs. Another example of a reservoir is the reservoir of Lyme disease. The white-footed mouse and other rodents, including wood rats and kangaroo rats, are all reservoirs, living reservoirs for Lyme disease. The Borrelia burgdorferi bacterium does not cause disease in those rodents, but the rodents can be a source of infection for other organisms. Interestingly enough, cholera has a human reservoir. The cholera bacterium lives in humans who don't experience disease from it, who are carriers of it. And then those people are a source of contamination for water and food and are the reason that epidemics of cholera occur. Now, I want to point out here that there's a difference between the term carrier and the term reservoir. When we talk about a carrier, we're talking about an individual. So, Typhoid Mary was a carrier of the typhus bacterium, but a reservoir is an entire group. Humans, in other words, can be a reservoir for cholera. It's important to keep those two terms separate. Another important source of pathogens is the vector. A vector is a living agent that can carry and sometimes help to maturity a particular pathogen and then transmit that pathogen to a susceptible host. Now, what I mean by helping to maturity is that some pathogens that are carried along in vectors are in an immature stage and they need to mature, to grow up before they become infective. So the pathogen is almost like an incubator. The vector is almost like an incubator for the pathogen in those instances. Vectors are almost always insects. They include things like mosquitoes and a whole lot of different flies. There are species of lice that serve as vectors, fleas and ticks and mites, all those little insects that nobody likes are vectors for sometimes very serious pathogens. Fomites are yet another source of pathogens, and all a fomite is an inanimate object that can harbor and transmit pathogens. So these are non-living things. These are things like surfaces, countertops, and workstations, syringes, dental tools, surgical instruments, nail files, nail trimmers, combs, and brushes, speaking of lice, doorknobs and handrails and light switches, computer keyboards, exercise equipment, even things like bedding and blankets and cardboard boxes. These things are capable of harboring certain pathogens, and therefore, when one person touches the fomite and leaves the pathogen behind, it can become a source for the next person who touches the fomite. Unfortunately, a lot of microbes can survive on fomites for an extended period of time, but other microbes are only capable of surviving on these inanimate objects for a limited amount of time. Now, what will determine how long a pathogen can survive on a fomite is typically whether or not that pathogen is alive, whether or not we're talking about a bacterial pathogen or a viral pathogen. So the key factors for a limited survival time on fomites are desiccation or drying out and extremes of temperature, either hot or cold. As we've discussed, living cells are quite susceptible to drying out. And so if the fomite is a particular surface or an object that tends to stay dry, that bacterial cell is not going to survive for too long. In comparison, some pathogens can survive for quite a long time on a fomite. There are particularly some viral pathogens that can survive for long periods of time on surfaces. A good example of that is the norovirus. There have been studies to show that noroviral particles can survive, if you will, for up to 4 weeks on hard surfaces and be transmitted to new hosts. Another scary example is the hepatitis C virus, which has been shown to be transmissible in pure water for as many as 7 days. Now, that data came out of a study of intravenous drug users who often share needles and share bottles of water to wash out their needles in between use. Once the bottle of water becomes contaminated with hepatitis C virus, the next time the person draws up water out of that bottle and into the syringe, they're drawing up viral particles for about 7 days. There's been a lot of interest in during the current pandemic as to how long the SARS-CoV-2 virus can survive on surfaces. And we don't have a complete data set yet, but it looks like it can survive for at least 24 hours on hard surfaces.